In this video, we will show you how you can share image analysis results with your colleagues using a web browser. You will see QuPath analysing whole slide images stored on a slide score server without downloading them and exchanging annotations and analysis results with the slide score server. All this works also for tissue microarrays. First, we need to enable QuPath integration for your account. You can see it's disabled as there is no Open in QuPath button next to the slide name. Click on your username, make sure the QuPath integration checkbox is checked, and click Save. A notification will pop up to verify. Going back to the study view, we see there's now an Open in QuPath button next to each slide. This button provides a link that can be opened in QuPath to work on a single slide. There is another Open in QuPath button under the study name to download a QuPath project file, containing all slides in the study so that you can run your analysis script over the whole study. Clicking the first one brings up a dialog with the link for the slide. Before we can use it, we need to install the QuPath extension that enables slide score data connection. Click the download link and drag the plugin over a QuPath window. A notification will show that the installation was successful. Now, we can go back to SlideScore and copy the link for the slide. In QuPath, open the File menu and click Open URI. Paste the provided link there, and after a short while, your slide will show up. You are now looking at a slide without having to download it. For this demonstration, we set the stain as H&E and detect cells in this region in the top of the slide. To do that, we will open the slide in SlideScore and use the Prepared Region of Interest annotation question. There is also a question to store our results from QuPath later. We will draw a rectangle specifying our region of interest. Press Done to close the annotation question and Submit to store the annotation. Now we switch back to QuPath and open the menu objects. There are now two commands to download and upload annotations to and from SlideScore server. First, we will download the Region of Interest rectangle. Make sure the Region of Interest question is selected and press OK. The rectangle that we drew in SlideScore in our web browser is now in QuPath. The next step is to run some simple analysis. We will detect cells using the default parameters within our region of interest. After a short while, we can inspect the results. In this case, we are not interested in the cell outlines, only in their positions, so we convert the cell detections to points. We can now double-click the 183 points annotation to select it, and using objects, upload annotations to SlideScore, send them back to the SlideScore server. This time, we select the cells question as destination. Note that the points will be represented as small circles. Reopen the slide in SlideScore and we can immediately see that the cells question indeed contains 183 shapes, our cells. Opening the annotation confirms they're in the region of interest. This video showed you how to enable QuPath integration in your account. Install the SlideScore plugin in QuPath, load annotations from SlideScore, analyze the slide, and send the results back to the SlideScore server to share with your colleagues. Try it at slidescore.com.